So, hello everybody on the screens, wherever you're in from. It's a big pleasure to welcome you to this webinar and panel session together with the three experts. Today we talk about antioxidants, a safe and viable inhibition strategy for nitrosamine formation in drug products. The session will be as such that we start with René Stemmler from DSM, talking about the basics, chemical topics and such. So to get you into the topic, then we have Kaushik Nanda, who will talk more about the formulation opportunities and the mitigation strategies. And last but not least, we will end the presentations with Malcolm Ross, talking a little bit about the regulatory, the challenges around, and then we go into a discussion with the three experts. Of course, in the meantime, you can raise your questions either in the chat or in the Q&A. And before to start really with René, I already have a quick question that I would like to raise. I hope I can do that.
Yes, it's on screen.
It's already on the screen. So the first question is, of course, are you already using any mitigation strategy? And it looks pretty head-to-head at the very moment. We have about 57%, 60% yes, 40% no. So it's going to be very interesting then to see the questions later on as well. So I let it run for another few seconds because I think people have made up their minds. So we have a 55% to a 45% no, which gives a good floor for starting this session. So René, without further ado, I would like to hand over to you just quickly to the ones that have not yet read who you are. René Stemmler is a senior scientist, process chemistry R&D at DSM. Huge experience in chemistry and the right person to kick this off. So the floor is yours. I'm gonna mute myself and take away my picture.
All right.
And we can see.
I understood that you can see the screen.
Yes.
Thank you, Philip. Hello everyone, and I would like to start off this webinar with a brief introduction to N-nitrosyl compounds and also their formation and their inhibition. So I will briefly touch upon the historical background. Is it a new issue? But also discuss a few vulnerable amines and also nitrosating agents that are commonly encountered, and also then conditions of how they form and how they could be inhibited. And I will close with a few mitigation options that one could use to prevent nitrosamine formation. So nitrosamines were first synthesized and described in the late 19th century, so quite a while ago by different chemists. And since then, there has been a nitrous acid test used by chemists as a qualitative analysis to distinguish the different amines, the primary, secondary, and tertiary amines visually. So have a simple test to understand what functional group they are dealing with. And for primary amine, this would be a gas evolution because it decomposes to nitrogen in the presence of nitrous acid. Reacting to different products, but the visualization is that the bubbles are formed. So you can see on the right side for the primary amine some gases forming. And for the secondary amine, this forms nitrosamine. This is the topic of today. And this typically is visualized by a separating yellowish oil phase that you can see in the middle. And whereas for tertiary amines, this basically stays a clear solution because primary, or initially at least, it forms a simple salt of ammonium nitrite. Back in the 50s, Barnes and McGee first reported that nitrosamine, specifically the N-nitrosyl dimethylamine, is a potent toxin in several animals. And since then, several more nitrosamine compounds have been identified to be cancerogenic. This list has been growing quite a bit. And then also in the early 70s, Linsky and Epstein then even proposed that these could be classified as environmental carcinogens because they occur in nature, and they also can form from secondary amines and nitrite in different matrices like food, water, cosmetics, and also especially in tobacco. And they can even form in vivo in the stomach. So this was the great concern in the 70s and 80s. So since then, this triggered quite a few studies on nitrosamines in general, and many specific compounds have been studied. I list here on the right, you can see a few structures of some of the prominent ones, but certainly this is not an exhaustive list. There are many more, but back then, those were at least the ones that were studied. So now fast forward to the recent years. Nitrosamine is a topic again in drug products, in drugs, and it started off with valsartan in 2018, when dimethyl and nitrosodimethyl amine was found in valsartan. And later on, several other sartans were also affected, where also nitrosamines were found. And this continued, and also in the coming years, that several more drugs were reported that also contained unacceptable levels of nitrosamines. And for the first two, at least, this is critical because these are part of the WHO list of essential medicines. So there are more than listed here. This is just a few examples. So what has been observed is that nitrosamines that were found often do not relate to the API structure itself. So often it is the case that impurities or reagents or excipients are in the manufacturing process introduce the reagents like the amines and the nitrite to form this nitrosamine. That means in turn that the nitrization can happen anywhere in the manufacturing process. Could start in the API production, but can also be in the formulation, could be in the packaging, or could be even on storage that nitrosamines still form. So this is basically over the whole process can be a problem. But also in the 70s, several scientists were also looking for ways to inhibit the formation of nitrosamines. So this was also a topic back then. And this is now something that we can take advantage of because there's quite some knowledge out there already about some additives and antioxidants that can inhibit the formation of nitrosamine, such as ascorbic acid, alpha-tocopherol, gallic acid, sulfide, cysteine, and others. And they do this by scavenging out the nitrite or the nitrosating agent, to be more specific, before it can produce the nitrosamine. So how can the nitrosamines form, or what is needed? So there are basically three ingredients or three conditions need to be met. You have to have a vulnerable amine that can be nitrosated, or an amide. You need to have a nitrosating agent present, and then the reaction conditions need to be favorable for this reaction to happen. Now on the left side, you can see different ways of how nitrosamine can be made chemically. But for our purpose, on the majority of cases, this is typically the first case on top in the red square by an N bond formation. So where typically a secondary or tertiary amine reacts with the nitrosating agent to make the nitrosamine. On the right side, there is a list of functional groups listed that are typically vulnerable to nitrization. So secondary amines and tertiary amines are probably well known by now, but also other functional groups, nitrogen-containing organic compounds, are vulnerable, such as ureas, amides, and carbamates, but also hydroxylamines, hydrazines, and hydrazides. Some are less reactive than the secondary tertiary amines, or than the secondary amines, sorry. But there are exceptions. So some of the ureas are also interestingly quite reactive towards nitrization. But these amide-derived structures are typically less stable, so they might not live very long, so they decompose quickly. On the other hand, the primary amines on the lower right are not considered vulnerable, because yes, they react with the nitrosating agent, but they quickly decompose to an azo compound and then release nitrogen and then decompose to other products. So they are typically not relevant. Then what kind of nitrosating agents are typically encountered? So the nitrosium salts in general are basically the most reactive nitrosating agents, but also nitrosyl halides and alkyl nitrites, and also the anhydrides of nitrosic acid, dinitrogen trioxide, and also the dinitrogen tetroxide are active nitrosating agents. But they're also precursors to that, so that's that can be contained in the drug product. And this is what most often is encountered is nitrite, as nitrite salts, but also organic nitro compounds can release a nitrosating agent, which is maybe not as well known, and other more exotic species of nitrogen compounds, such as Fremius salts, can also be a nitrosating agent, or can become a nitrosating agent. So what reactions conditions are needed or are favorable? And there are indeed quite a wide range of reaction conditions that are known to enable or allow nitrosation. And you can see on this scheme 3 columns for the different media. So we have aqueous solution, the gas state, and non-polar media. And on the y-axis, the oxidation state of the nitrogen of the nitric oxide species. And you can see in the gray square is the area where the nitrosating agents are used typically. So these are active nitrosating agents, typically with the oxidation state +3 or +4 of the nitrogen to take part in the nitrosation. And outside, this is not a nitrosating agent, so nitrogen oxide and nitrate are not per se nitrosating agents, but they can become nitrosating agents. For example, the nitrate can be reduced, for example, by microorganisms to nitrite, which then in aqueous solution, at least, and especially in acidic conditions, transforms into nitrous acid, which then can lose water and convert to dinitrogen trioxide. So these species then are all active in nitrization, and they can also cross the media, so the matrices. So it could go from the aqueous solution into the gas state, and also could migrate into a non-polar media, meaning that it's not, it could be happening in different matrices. Now the nitrate or the nitrosating agents can also be converted into nitrogen oxide by reduction. And there are different agents then that can do that, basically converting this into a non-nitrosating agent. In aqueous solution, typically or often or very prominent being used is ascorbic acid. In fat-soluble media, it's typically can be used alpha tocopherol that can do that as an example. But also in the gas phase, sulfur dioxide could reduce these species to a non-nitrosating agent. And this is specifically the mechanism by which these additives, these antioxidants can scavenge or remove the nitrite or the nitrosating agents from, and keeping them from nitrosating amines in the drug product. So what options could one do, assuming the risk assessment has been done and it came up that there's a risk that the drug product or can form nitrosamines. This is not an exhaustive list, but certainly a start, and also part of the of a suggestion by the FDA that that this could be used. So the first one is, of course, the more obvious one or the laborious one, to ensure that you use reagents that are free of nitrosating agents and their precursors, maybe ideally also free of amines that could be nitrosated as far as possible. But this is it's laborious, and you cannot always prevent that. But also nitrization may be inhibited by certain conditions, so especially in basic pH, which could be maybe helped by addition of sodium carbonate as an excipient to move the microenvironment to basic pHs where the nitrization is at least strongly reduced in rate, so slowed down quite a bit. And there's some evidence that this can indeed help in certain cases. A more versatile and more broadly applicable option would be to add antioxidants like ascorbic acid or tocopherol to basically scavenge any nitrate that could be present in your product. And this is also a mitigation strategy that is recommended by the FDA. So with this, I'm at the end of my talk, and thank you for listening.
Thanks a lot, René. Very nice insights into the chemistry and into the root cause, and already touching some points of potential mitigation strategies, for which I would like to hand over more now to Kaushik Nanda. Kaushik Nanda is a pharmaceutical scientist at Merck and has a lot of experience also in drug degradation chemistry and the like. We had a chat before we started the webinar, and I read also your latest publication on mitigation strategies. So I would like just simply to hand over the floor to you, Kaushik, and looking forward to your presentation.
Thanks, Philip. Can you see my screen?
Yeah.
Perfect. So, thanks to René, because you basically set the stage, and I do not have to spend much time on the chemistry of nitrosamine formation, which I already had on my slides. What René told you, an exhaustive list of chemical conditions which may form nitrosamines. Here, what I am showing, it's a condensed from the most probable space where nitrosamine may form. So for formulating a drug product, when you have an API, most of the time, the most vulnerable amine we know, it's a secondary amine. If you have a tertiary amine somewhere there in your API or as a impurity, they also may form nitrosamines, but the rate of formation is much slower, so the risk will be much slower. But if you have a secondary amine, it can form nitrosamine quite easily, and as René mentioned, under basic pH, although under acidic pH, although under basic pH also you may form it, depending on if you have a bit of aldehyde around or so. There is a mechanistic pathway for that. But for most of our purpose, for 99% of cases, if you have a secondary amine, nitrite in yours and nitrite, a bit of basic pH, then it's a problem. Now, when you form nitrosamine in drug substance while you are manufacturing, and if you are aware of it, you can always change your process route, or you can change the purging factor, so to speak, in such a way that the amount of nitrosamine will be below acceptable range. But if you start forming nitrosamine in your drug product, you cannot purge it. So that's where the mitigation strategies come. To mitigate nitrosamine formation in drug product, as by now you know, there are two main ingredients, a vulnerable amine and a nitrite. Now, nitrite itself does not react to form nitrosamine. it has to react with most of the time with acid to form the nitrosating agents, which Rene listed. In here, I just listed two most common ones, NO plus or N2O3. In our studies, to show that we can really inhibit formation of nitrosamine in drug product, we try to exploit these three mechanisms. The first mechanism is redox using an antioxidant. In this case, this is ascorbic acid. So ascorbic acid reacts with the nitrosating agent and basically it neutralizes it. Or what you can do, we can use a diazotization pathway. Instead of an antioxidant, you can also have a primary amine containing inhibitor, which will neutralize nitrite. That means it won't form, there will not be enough nitrite to form nitrosating agent and hence nitrosamines. And another mechanism also we exploited using inhibitors, basically polyphenolic inhibitors. Polyphenolic inhibitors can react with nitrite and basically they themselves get nitrated. This is how it removes nitrite from the system. So to show the proof of concept that we can really inhibit nitrosamine formation in drug product, we chose this amine. It's a model amine. It's not a drug candidate. But if you look at this structure of this amine, secondary amine, it is not only a secondary amine, it's a cyclic. This secondary amine is in a cyclic system. It's a piperidine. Usually, these cyclic amines are more reactive or more nucleophilic than a linear secondary amine, a non-cyclic secondary amine. So what we did, we formulated this drug, this model drug, with some common excipients. We used microcrystalline cellulose, lactose, sodium starch glycolate, silicon dioxide, and magnesium stearate. We formulated them as 100 milligram tablets, and some of those tablets were not spiked with the inhibitors, and other tablets were spiked with these inhibitors: ascorbic acid, sodium ascorbate, vitamin E, ferulic, and caffeic acid. Once they're made, what we did, we stressed those tablets at 50C 75% RH for 1 month. Please note that this 50C 75% RH is not a usual stress condition we use. It is very stressful, this stress condition. But we wanted to get data sooner than later. So instead of waiting for 6 months under a lower stress condition, we used this unrealistic stress condition to show that we can still inhibit nitrosamine formation. So what happened? Tablets containing no inhibitor in this 1 month under these stress conditions formed 345 parts per billion nitrosamine, normalized to the tablet weight. In 100 milligram, 345 parts per billion nitrosamine we found. Now, these inhibitors, we used 2 levels. One is about 0.1%, not in this case, not about. For ascorbic acid, it is 0.1 weight% and 1 weight%. There are 2 inhibition spike levels. Under these spiking levels, what we found is for all these inhibitors at 1 weight% spiking, the inhibition efficiency is greater than 80%. We calculated if you get 80% inhibition of nitrosamine formation in a solid doses form for most, if not all, drug product, taking into account how much nitrite you get as impurity in the from the excipients, you will be below the regulatory acceptable daily intake of nitrosamines, which is 18 nanogram for nitrosamines for which there is no toxicological data. And for other nitrosamines like NDMA, NDEA, this level is much higher. So even when the level is only 18 nanograms, with a 80% inhibition, we can get below the acceptable daily intake of nitrosamines. Well, for your information, we also did some work using exploiting the diazotization pathway. So for that, we chose 3 amino acids where it has free primary amino group there. And all these 3 amino acids, they are from FDA's inactive ingredient list. That means you can use it if you want to. So it's in solution phase, we did some, ran some model reactions, and we used pretty stringent condition, pH 3, where the rate of reaction for nitrosamine formation is fastest, 60 degrees Celsius, pretty high temperature, and we gave about 1 day for the reaction to happen. Under the stringent, even under the stringent conditions, we found that all these 3 amino acids, they did good work in inhibiting nitrosamine formation. And when we used histidine, it was above 90%. It's pretty good. That means we can say that now, after looking at this data, inhibition of nitrosamine formation can be achieved via multiple mechanistic pathways. One of them is antioxidant, like ascorbic acid, sodium ascorbate, or other antioxidants, which we have not shown here, could be used. We can also exploit the diazotization pathway. We can add primary amine containing inhibitors to stop formation of inhibition, and that can happen both in solid state and also in solution state. So if you have a drug product which is injectable, which is a suspension, you can use this similar strategy. Although I'm saying in our study, in this work, we are showing this histidine, these amino acids only in solution state. It does not mean that you cannot use ascorbic acid there. You can use antioxidant also for your injectables or suspension drug products. With that, I'll stop here and I'll take any questions.
Perfect. Thank you so much for this nice overview about now coming from René's point of the topic itself. Now coming over to you, Kaushik, that shows potential possible mitigation strategies. Now, of course, comes the question to the audience right away before we go to Malcolm. Would you use such a mitigation strategy in your daily life, in your development work? So I'm happy to take your votes also here. The yes is popping up logically, I assume. I would have been surprised if it would be anything else than this, because we need to find solutions. We need to find the right solutions. Okay. And I thank everybody for participating. So we are a 96% yes to a 4% no rate currently. And of course, we will take your questions both or for all 3, René, Kaushik, and Malcolm, after the presentation by Malcolm, because now we come to the different topic. It's, we have solutions. Might there be challenges? What can be situations on a regulatory level where Malcolm Ross will dig into his speech? And Malcolm himself is president at General Pharma Training and Consultancy. I met him first time in my life at a pharma lounge in Basel, where we talked about cultural differences in the pharmaceutical industry. very nice presentation and talk about also there. And I cannot wait to let you go on stage and hear what you have to say. So title, nitrosamine prevention, the challenges.
Okay, so it would appear that we have solutions. We understand the problem. We have potential solutions, but these solutions, unfortunately, may come at a price. And just a historical comment, as usual, the pharmaceutical industry is the last industry to wake up to the fact that there is a problem. We see it in 2018, the brewing industry have been aware of NDMA in beer since the early 80s, and they actually solved the problem by changing technology. Most of the problem with the nitrosamines was coming in the way that they heated the kilns that were used in the malting process. And most of the food industry, meats, particularly processed meats, the rubber and tire industry with vulcanization, the cosmetics industry, they've all been looking at this problem for 30, 40, 50, even 60 years. This has been known, and we've only just woken up to it. So partially, I think we're a little bit to blame for the panic situation that we're in now. But of course, if you don't look for something, particularly at the PPP level, then it isn't there. And I think that's the approach we adopted. So can we learn anything from these other industries? And I think we can. One of the things that I think we can learn, I see in many of the FDA suggestions, the EMA suggestions, what about getting nitrite-free excipients? If you look at this paper, which was published a long time ago, nitrite curing in meat, if it was possible, the most reliable method to alleviate the N-nitrosamine problem would be the total elimination of nitrite from the curing process. Of course, a whole new set of problems that arrived, and basically they're talking about the fact that if you tried to get rid of nitrites, half your products in the food industry would just disappear because it can't be done. And I think that I learned from this, that this discussion that's going on about nitrite-free excipients is simply an unrealistic approach. I think we will continue to live with nitrite in excipients. If you do go to an excipient manufacturer and say, can you manufacture for me a nitrite-free excipient? The answer will be yes. How many thousands of tons do you want? Because these excipient manufacturers are manufacturing for the food industry, not for the pharmaceutical industry. This discussion about GMP for excipients has been going on for ages, and it's the same argument that you would have here. So what are we really looking for? We're looking for a magic wand. So I created a perfect solution, nitrosamine expelliarmus. And what are we looking for? The perfect prevention of nitrosamine formation. The additive should require no change to the manufacturing process. It shouldn't affect the critical quality attributes of the product. It shouldn't affect the appearance and organoleptic qualities. It should be acceptable to the regulatory authorities with no requirements for any additional information beyond proving effectiveness of nitrosamine prevention. So it would be a type 1 do and tell or annual review variation with no extra requirements. That would be nice. However, in the same way that Harry Potter is fiction, so is those requirements when you come to looking for a solution. Ascorbic acid, the subject of today. It's got a lot of advantages. First of all, it works. We've seen this already, examples which show that it works. It's an approved pharmaceutical substance. It appears in the FDA non-active pharmaceutical ingredient database. It's generally recognized as safe. A good point. It is available in pharmaceutical quality. There's USP, BP, EP. It's a standardized material. Some of the other suggested scavengers, the polyphenolics, the plant extracts, there's quite a lot, if you look in the literature, the use of polyphenolics, flavonoids from plants, they may well work, but they're not standardized materials. So you would have quite a long path to get these qualified. It will appeal to regulators because it's something they're familiar with. It's recognized, it's used as an additive in many formulations, it's used as an antioxidant. So I don't think any regulator is going to raise their hand and say, oh, what about toxicology, etc. It's an easy win. There may be some additional possibilities with ascorbic acid together with tocopherol or citric acid. There are a number of suggestions, even some patents, suggesting that combination of ascorbic acid together with other materials such as tocopherol or citric acid may actually augment the activity as a nitrosamine preventer. The con side, you'd never get anything for free. We must always remember that depending on the amount of ascorbic acid that is required, this may alter the properties of your processing. So you may have to reformulate. Obviously, if you're developing new products, that's not a problem. But many of us are suffering from portfolios of old products which are now at risk. As we know, ranitidine was completely at risk and taken off the market. But there are many other APIs that are coming along, old products, known products, which are now shown to have nitrosamines, valsartan being the first. It may, depending on the amounts that you need, affect the overall processing, the formation of a tablet or something like this. I would remind everybody, ascorbic acid is not a chemically inert molecule. It is reactive, and it can react with different mechanisms with the drug substance itself. So once again, you are going to have to look at this on a case-by-case approach. Ascorbic acid is not a placebo. It has its own chemical properties. And unfortunately, from my interactions so far, I don't think we're going to get any freebies here from the regulatory authorities. If you are putting an additional material such as ascorbic acid, yes, the toxicity will not be a question. You will be required to prove effectiveness in preventing nitrosamine formation. And in spite of the experiments at 50 degrees centigrade, I think that's going to have to be a long-term stability, 6 months accelerated and shelf life. So that's 2 years plus before you get to market with a reformulated product. You will almost certainly require new stability data because, as I say, ascorbic acid in itself has the possibility of chemical pathways. And probably the biggest downside, because all of that we can cope with, we know how to do it, we know how much it costs, we know how long it takes. I am concerned that for solid dosage forms, we may well be asked to repeat bioavailability. I believe that we should be hoping that the regulators will allow us bio waivers based on comparative dissolution data, the product with ascorbic acid and without ascorbic acid. And certainly for BCS class 1, class 3 products where you can even get bio waivers without that sort of argument for generic products. But I suspect that for modified release, if I read the guidelines correctly, a change in formulation, an addition of a new excipient, unless you have a fantastic in vivo, in vitro correlation, which you probably don't have for older products, you will be required to do another biostudy, even if you have comparative dissolution data. That's the way the guidelines read at the moment. So my message here is that ascorbic acid may work, not for every product. It may create problems with other products. It's the best and most obvious way to go at this moment, because if you look at the pros, it's going to be acceptable. But we may still have to be paying a regulatory price from most authorities who will be demanding certainly new stability data, both in the prevention of nitrosamine formation and in stability of the drug substance itself. And the bioavailability could be a major cost when we look, and I'm looking very much from the generic point of view. During the development of a new product, that's a natural price you pay. But from a generic product where you have companies with hundreds of products, if not thousands of products, of which maybe 5% fall into the category of APIs or API impurities that could potentially form nitrosamines, this is going to be a huge burden on those companies. And I personally don't see how it's going to be dealt with. That's the end of my presentation. And
Thank you.
I'm very interested, particularly from those companies who've got these hundreds of products, to hear how they're planning to deal with it.
Absolutely. So thanks a lot to all of three of you. I think it's a great overview that we got from the problem to the potential solutions, or the solutions that we can, apart from mitigate them right away, to potential challenges. And nevertheless, I like to take up challenges or questions. And one was mentioned by you, Malcolm, on the different amounts. I think it's inherent in every formulation that there is, as I used to say, there is no formulation for everything. But Kaushik, I would like to come to you on that question. Do you see with the low percentage, now purely talking technical and processing, the low percentages that you mentioned below or around 1%, do you see an impact, a critical impact on the manufacturing process itself, or would you consider this like neglectable more?
There will not be a major impact on the manufacturing process, but depending on what is your API, is physical characteristics,
yeah,
and what are the excipients you already have, you may have to, it is not like just sprinkle 1% ascorbic acid powder on it, right? So you may have to change, and we have done few of those things in our shop. You have to change the process a bit.
Okay.
But it should not be a major challenge, though. It could be done. Okay.
And yeah, it goes in the direction. I think with new formulations, you have, in any case, you have to do your process development, your DOE, your stability testing. So we don't have to talk about the new developments. I think it's the challenge for existing products, like big products like metformin that you mentioned, the sartans that were mentioned. And then, of course, you also mentioned, Malcolm, the non-inertness of ascorbic acid. Of course, if it prevents something, it can also act in a different way. Now, René, for this Maillard reaction, are there limits, or how do you chemically, is there a possibility to say, there is a certain amount that prevents, and up from a certain amount, it goes in the wrong direction? Or how would you see that from a chemical point of view?
I think from the chemistry point of view, it's certainly a matter of rate. So with what can ascorbic acid react first?
Yeah.
If we're targeting nitrite and assuming that this is captured, this will certainly react first with the nitrite. But if you have excess, of course, this could find other partners to react with if there are reactive partners around. So certainly this can happen. If you have amines present to have the Maillard reaction going, then this can happen. But I mean, at the levels that Kausick presented are maybe comparably small.
Yeah.
And maybe the effect of the Maillard reaction is then minor.
Okay, so use them all to avoid the nitrosamines and avoid further reactions. This would be, if I summarize in my simple words, this would be the way forward that you would see, right?
Yeah, and in addition, it does not need necessarily to be only ascorbic acid, or it doesn't need to be ascorbic acid. I mean, Kausick has presented several options, and there are even additional options to mitigate. Ascorbic is certainly the one of the best ones, the best studied ones, and it works very well. Also, Kausick has shown that again. So this is maybe one of the options to go for, but it is not necessarily the one.
Okay, if I understood correctly, the ascorbic acid is well approved and has the regulatory approval, so it can be used, and others that potentially would work as well are not. Coming to that point, I'm not sure to whom to really put this, because it is in the use. There is a question from Fabrizio: is vitamin E TPGS equally effective as vitamin E for reducing nitrosamine formation? Not sure, Rene or Kausick?
I'll take it. Being a scientist, I do not have the data, so I cannot say 100% it will be similarly effective. But again, being a scientist, when you look at the mechanism, it will be effective, similarly effective.
Okay, okay. So we have two very viable options here, I think. I would love to come back to Malcolm's point, and this time going to Malcolm. You mentioned the topic on the regulatory, right? How it will be accepted, what will be the outcome. Can it not be that it will also strongly depend on the need for those products, how regulatory will react on proposals done by the pharmaceutical industry? I just ask this because I saw in our COVID crisis that excipients that are now in vital injections, as we call the vaccines, were not approved beforehand, but they went through like a charm. So how do you see? Because when I see metformin is one of the most important products in diabetes treatment as well. So how do you see the chances there that regulatory authorities act a little bit more nicely?
Well, I'm answering this in my own name and not with any of the companies that I am affiliated with.
Okay.
Up until now, I have not seen any indication that the regulatory authorities are demonstrating any sympathy for our problem or any logic. We all know that we swallow more nitrosamines in our food on a daily basis, far more than the levels of the 18 ppbs that they're allowing for some of these products. We have made the argument that we are taking in cooked meats, in cheese, in beer, in whiskey, and the answer has come back, and my colleagues in Novartis have basically told me that they've given up the argument. The argument comes back from EMA, yes, but it's a life choice if you decide to eat cheese or not eat cheese, but you don't have a choice about whether you take drugs. And we are looking at, and correct me if I'm wrong, On a normal diet, we're looking at factors of maybe 100 times more daily intake of nitrosamines with a normal diet than we could ever get from pharmaceuticals. And again, with some of these things like valsartan, lifetime, we eat for the whole of our lifetime, but you don't start taking blood pressure drugs until 50 or older. It becomes illogical. I've seen no indication of the regulatory authorities making any adjustment or move on this. If anything, they are getting more intractable. Now, I will give one extra point. There was recently a presentation from somebody from the TGA, the Australian authority, where they did mention, and I think it's in the regulation, if banning a particular product results in drug shortage, and they gave the example of metformin, said if there was no metformin, nitrosamine-free metformin, there is, but if it proved that there would be no availability of nitrosamine-free metformin, the risk of not taking metformin because it's a first-line treatment for diabetes, the risk of not taking it would be sufficient to outweigh banning it. And so there is an understanding, but we haven't yet had one of those cases, because the sartans you could get with low or free nitrosamine. Metformin had products on the market. Ranitidine, there was an alternative therapy, practically equivalent, but it will come to the crunch. There are several things in the pipeline where nitrosamines have been discovered. It's something in a wide range of products that could result in people being unable to get their drugs. But we'll see.
It says it's all. It's going to be kind of a political game.
It's very much, it's pharmaceutical with a big P for politics.
Exactly. So, but I would like to come back to some of the questions from the audience, because we have plenty of those. And don't worry, the ones that are not answered now, they will be answered later on. But I would like to come back to some also on the formulation, and this goes to Kaushik. How do you ensure homogeneity of these antioxidants in the formulation? Pretty simple question might be, but perhaps it's not so easy.
Well, depends on how you do it. Suppose, because if you are doing a 0.1% spike, which is very small, so it has to be homogeneous. So what we did in our model study, we basically made a solution of ascorbic acid and did weight granulation with a excipient we are using, which we are using in a larger amount. And then we controlled the amount of antioxidant by controlling that spiked excipient.
Yeah, because you want to have it everywhere.
Right.
Whereas the API, you have to have the ascorbic acid nearby, otherwise it will be concentrated.
This is very important. That's why weight granulation or procedures like that will be preferable most of the time.
Very good. So now I'm not sure if it's René or Kaushik to answer that one. What is the most critical factor regarding nitrosamine formation? Temperature, moisture, or both combined? Are dry processes considered as safe?
René, once you are, I can take this question, because we have done some work in the formation also, because at the beginning, when we had to do risk assessment of all of our marketed product, we had to generate data to understand how nitrosamine form in drug product.
Yeah.
Just to answer that question, whether the dry process is safe or not, it is not. It is in terms of forming nitrosamine while processing, it is safer than the wet process. But after you process, you make your drug product, when it sits there on storage, there is no difference between dry and wet granulation or wet process, unless under wet processing, you are destroying the crystallinity of your API.
Okay.
If you make it more amorphous, it will react much faster. So it depends on quite a few factors.
Yeah,
Okay.
if I can add to that,
Sure.
in our initial assessments, we said that dry processing, direct compression, no risk, and that was our original concept. We are now saying, exactly as you said, less risk. We have seen a product which is a completely dry process, and yet nitrosamine is formed during the process. We don't understand why, but it's currently we're looking at root cause. But I'll add an additional one. We actually have a drug substance which, if you leave the drug substance on the laboratory bench, converts to a nitrosamine from the nitrous oxide in the air. So even air, and of course, when you're looking at things like fluid bed drying, or you're looking at tablet coating, for example, you are punching a hell of a lot of air through your product, and that air contains nitrous oxide, which you can check. You can buy a nitrous oxide detector, very cheap, and you will be quite surprised what could be happening. Even to the extent that I've heard discussions that we may need to put catalytic converters on the inlets of things like fluid bed dryers.
Oh, wow.
So every day is a new surprise when it comes to nitrosamines.
Yeah, use the inhibitor, makes life simpler if you can.
Sorry, say again?
Use the inhibitor formation.
Yeah. I mean, the technological solution of getting a gas, an air supply that goes through a filter, catalytic filter going into a fluid bed dryer for the volumes of air and the speed, this would mean basically reconstructing the whole of your factory.
And it does not solve the problem of forming nitrosamine on storage.
Exactly, you're absolutely right. But just to say, the question is, what are the factors? The factors are anything that they feel like being. I think the only factor that we can put our hands on our heart and say is pH is probably the most critical of the factors, but it's certainly not that alone.
Oh, okie doke. So coming back to square one, this brings us really, because if we cannot close out the sources to get the stuff in, we have to do something to keep it low or to have a mitigation strategy to avoid it. You mentioned the measurement. I think it was Kausik in his presentation. How did you, which technology did you use to analyze the nitrosamines?
It is a well, this is a challenging one because you are trying to go to the detection quantitation limit of 10 to 20 parts per billion, which is, so what we used, it's a LC-MS technology.
Yeah.
It's
okay.
a LC, but we quantify with MS.
Okay.
Mass spectrometry.
And did you perform any interaction studies of the scavenger and model compound API? Well,
for this particular one we used,
okay,
there is no interaction. We know.
Yeah,
we have checked it. But as Rene said, you can have another API or another impurity which will have some part in the molecule which can react with which can go through the redox process in which ascorbic acid is involved.
Okay,
that is possible.
And then another question, but this goes now to Rene. You mentioned that FDA has recommended using mitigation. Did that recommendation include scavenger in drug products? And if so, would it be possible to provide a link to that reference?
So what the FDA is mentioning on the website is that it's posing a potential mitigation strategy. So they mentioned this mitigation strategy specifically.
Okay.
So I can post a link right in the chat right now.
That'll be cool. That'll
Just
be cool.
one thing here to mention. I mean, in that communication from FDA, they basically mentioned our publication, the work for what I presented here today. That's the mitigation strategy.
Yeah.
So in these slides, there is, I think, not last slide, last but one or so, there is the reference of our paper. Or in FDA communication also, there will be reference.
I think it's important to understand that when they use the word recommend, it doesn't mean it's going to work. It means recommend to try.
Yeah.
Agreed.
And so please don't overinterpret it and think that the FDA has said adding ascorbic acid will solve your problems.
Absolutely. It's just a recommendation to try it, right?
Yeah.
Okay. So we have used our time together. I would love to continue talking about all the topics on the nitrosamines, how to prevent them, and go further. But time's up. This leads me to thank everybody that participated as a listener in this case, or watcher interested in the topic. I would like to really thank all three of you for your very valuable insights on this topic, on this very important topic. And I'm sure the discussion will continue. We will find out more and more over time, and perhaps there is even the occasion to do another session on this topic. So thanks everybody, wherever you are in the world, either a good afternoon, good night, or still a good day in front of you. With this, I would close the session. Thank everybody again and wish you all a good time. Bye-bye.
Thanks, Philip. Thanks, everyone. Bye.
Thank you.
Bye-bye.
Bye-bye.